Okay, so this next piece, last piece, super important, but also really weird. So 100% you need your notes. You're probably going to want to go back and listen to this a few times. You might want to go either into the slides and dump pictures into your notes, or I guess take screenshots of this and dump them into your notes too, whatever you want to do. But you definitely want pictures. You definitely want notes. This is weird. So we're going to talk about making things rational, like pun, math class, fractions. So dilutions can also be expressed as something called dilution factors. Super important. You will see this on upcoming tests and quizzes. You will see this on NOCD. So the reason why is this is really, really important in terms of a biology lab and in terms of microbiology labs, which is where this kind of image is coming from. We'll talk about how this plays into microbio and virology in a few slides. But first, we're just going to talk about what a dilution factor is. So the dilution factor is always written as 1 in the numerator. So it represents one part of what the original is. And the denominator -1 is the parts of water. So it's kind of like with respect to the original stock solution. So if we look here, we have a bacterial culture. And I take 1 milliliter out of here and I add 9 milliliters of water. So it is 1/10 of the original concentration of your solution. So I had 1 milliliter of the stock, 9 mils of water. So it is 1/10 of what it originally, like the strength of that original stock solution. From there, because it's a serial dilution, so I keep taking from the thing that I just made. So I'm going to take 1 milliliter out, 9 milliliters of water. So this is 1/10 of this, but that means it is 1/100th of the original stock solution. Get it? So it's a 1 to 10 and then another 1 to 10. So that's like a 1 to 10 of this, but a 1 to 100th of this. And if I take another 1 to 10 of that, that's 1 to 10 of the previous, 1 to 100 of that first diluent, 1 to 1,000th of my original stock solution. So it's always 1 because I'm taking like one part out and then the total parts that are there. So it'd be the 1 part from the solution and then 9 parts extra of water. So that y would be 1/10. So if kind of how this is going to play out, we're going to talk about this more, but notice here that those broth then are getting inoculated onto Petri dishes. This is done to talk about or to determine the strength of the colony or how virulent a virus is. So when we talk about how we hear in the news about, oh my goodness, this strain is more virulent, this strain is this this is how it is traditionally determined in a lab setting. So you're going to, not viruses, we're talking bacteria right now, and we'll talk about why that's different in a second. So we're going to plate those broth on these culture dishes. And when it is so strong, so your stock solution, there are way too many colonies to count. Like you can't count individual bacterial colonies. Here, I plate it, I let it grow, I still can't count it. There's greater than 500. There's no way that I am sitting there and counting that many colonies. I pour this one. So this is 1 to 100th of my original stock. Okay, there's 71. That's better, but that's still a lot of counting. Here, over, so this is 1/1000th of my original stock solution. I have 6. Oh, I can count that. 6 is a nice number that I can count to. Over here, 0 colonies. So what are you counting? Nothing. So this is that it is too dilute to actually make it, that there's no colonies in there. This we would say is too few to count. Too many to count on here, too many. This is too few. It's not going to give you enough information. So when we're looking at this, we perform a serial dilution. Okay, so that serial, that it's 1 part, and then we fill in with the water. We keep using the previous one to get to a lower amount, to get to a more dilutant version. And what we're doing is we're determining the strength of a bacterial culture from this. So like we said, recapping here, big things here. We've got dilutant factors. Dilutant factors talk about the power of the solution. They relate it back to the original stock solution. So when I took here, I had 1 part of that stock, 9 parts of water. So this is 1 to 10 or 1/10 of the original stock solution. From here, I took 1 milliliter out, 9 mils of water. So this is 1/10 of the previous one, but it's 1/100th of the original stock solution. Okie dokes. So, like I said, it is weird, but like, look here. So if I did this, this was a 1 to 5, and then I did a 1 to 10, it would be the same as doing a direct 1 to 50th dilution. Okay, so think about here. If I had a concentrated stock and I do a 1 to 20, and then a 1 to 2, then a 1 to 100. If I want to know what my final one is in respect to the original stock, I multiply all those together and I get my answer. Okay, same kind of thing here. So if I do 1 to, if I'm talking here, notice this is 1 mil stock and 49 milliliters of water. So this is 1 to 50. This is a 1 to 50. This is a 1 to 9. So you would have 50 times 50 times 9 to give you the final diluent strength that's going to be there. All right, so here is just another picture. This is a great picture, really, really awesome to be putting into your notes. So these are what we call colony forming units, CFU. So we do our serial dilution. Okay, so we do a 1 to 10th, a 1 to 100th. Okay, so we're talking about all the different dilutants increasing by a power of 10 each time. Notice here that it's talking about too many to count. This is what they're going to take from. Really, 2 is like, come on now, you can count higher than 2. 17 is probably where I would go, not better number, because 160, I'd be afraid that I was going to lose one. But if we have, there's also these machines that you can put a plate under and it will like read the plate and be able to count this for you. But like in old time, like you used to like have like a paper and like a quadrant paper underneath, and you like literally would like count how many colonies were there. It was terrible. But here we have, so it was 159. It's a 10 to 3 dilution. So that means the colony forming units, the CFUs, if we do 159 times 10 to the third, is 1.59 times 10 to the fifth. So scientific notation, status happening. And this is your CFU. So this is what they're talking about, is the strength of that original culture, the strength of this original stock that's up here. It has 1.59 times 10 to the fifth CFU. All right, so how they do it with viruses. So viruses, very similar, but we're talking about plaque forming units, so PFU. So where this, when we were looking here, we were looking for CFU, the colony forming, so like how many colonies are present. Here we're looking for how many plaques are present. And the reason why, I'm going to come back to this. I want to show you, I want to show you this video first. Come on. There it goes. So what happens when you have a virus? Do you see how like it's like spreading out from the middle? What that is it's creating what is called a plaque. And as what viruses do is they kill the cell. So when you have a plaque, what that really is, it's an area of dead cells that are there because the virus killed them. So now going backwards, okay, so now we have my virus stock. And again, same thing. I want to know how strong my virus is, how virulent my virus is. I'm going to do a serial dilution. Okay. If I look at this, my 10 to the 5th dilutant, it's way too many to count. 10 to the 7th, come on now, you can count higher than 2. 17, that's a nice number here. So here I would have 17, so 1.7 times 10 to the 1 milliliter times the 6 times 17. We've got 10 to the 8th. or 1.7 times 10 to the 8th plaque forming units. Because I'm going to multiply this by this by this and get my PFUs. Now, why that's bold like that, I don't know why, but this is called a virus titer. So it's looking, like we said, looking to determine the strength of the virus, or how virulent, how aggressive this virus is. The way that they do it, let me scooch me over. So the way that they do it is they put the cells down, and then they actually put a kind of like an agar on top of it, and then it kind of will like eat through. So as it goes, it will change and morph the shape of the cells, but it's done with this like nutrient agar on top of it. Because, so we have like a lawn of cells, and you can see that's where the first infection was. As that spreads, that's what you have, this plaque. So not all of them are going to produce like a zone of dead cells. Sometimes they can be treated to have a color. And so instead of looking for like these clear areas, you're looking for a colored area. So here we have one that is expressing like a blue pigment as it kind of lyses those cells open or kills those cells. All right, so here's this video that we watched again. So what Penguin is asking you is what would happen if instead of having agar over, we actually had just a buffer, like just liquid over it? How would the plaque differ? So if this is what we're looking for with plaques when they have agar laying over them, what do you think would change if we just had like a liquid floating over this area? Like how would that differ in terms of how this plaque would present? What do you think? Okay, so here's the next big thing. What we're going to do is we're going to combine everything together. Super, super, super important. Okay, big thing here. Penguin, look, Penguin's in a ghost costume. Spooky, you need to pay attention to this. What we're going to do, come on, I'm trying to, all right, fine, whatever. Fine. All right, so what you're going to do, you're going to be making a graph here. So here's your first set of known absorbances and concentrations. So you would make this graph just like you have been making your 3D, your E, and your F graphs, and your copper sulfate one. So kind of think about it like you're plotting these 3 points. Then you're going to use your least squared regression line to help you. If I give you these absorbances, can you determine what the unknown concentration is? Point of kind of like clarification here. When you have the brackets like this, that means concentration. So when you have unknown concentrations, so if I give you these 2 absorbances, the Y, can you determine the concentration? The X. Then from this first one, okay, once you determine that concentration, you are going to perform a 1 to 4 dilution. 4 is the denominator, so that is the total change that is happening. What would the new concentration be? Big things. Make sure you're okay with this. Make sure if you're not that you ask questions when I come back, because this will come back to haunt you. Guaranteed. Make sure you're okay with this. Okie doke.